Thank you for. Does this thing work? Can you hear me? Thank you for having me. Thanks to the organizers, Shreya, Guillaume, and Meming, and to the Champalimo Foundation. I'm very excited to be here and hopefully, interact with many of you the rest of the week. So I would. Does this work if I'm away from that? Yeah. I guess the feedback is so quick that I can't hear myself. I want to talk about non-canonical substrates of biological computation. So I want to cover a few different topics in this talk.
Let me just see that.
I can just use these actually.
You want to talk with those?
Yeah.
Okay.
Sorry. Okay, thanks. So the running theme is that we're trying to consider unexpected ways in which the components of the nervous system can talk to each other and interact with the world and the body. So I want to start with a simple cartoon explanation of one problem that biological systems face, animals, as was already discussed before by the organizers, which is integration across systems and time, and more broadly, a control theory of how animals interact with the world. So imagine an animal in a forest wanting to eat grass. So if this is a herbivore, it'll also know that there might be predators hiding in the trees. Now, if it moves forward to eat the grass, it'll be more visible and more vulnerable as a consequence. So the brain has to decide, has to weigh the drive to eat versus the drive to stay safe. So eventually the brain will make this decision, but based on many different pieces of information from different sources. The initial hunger signal will come typically from distributed systems across the body. That needs to be combined with memory of what the animals previously experienced in this part of its environment, which then has to be integrated with new information from vision, olfaction, and audition, for example, which itself is integrated over some period of time as animals make observations over a period of multiple seconds about the current state of the environment. That then has to lead to a decision of whether to move or not, which has to then be executed through the motor system. And all these functions engage different parts of the brain. And this is one reason to study the brain as a holistic system. Imagine the animal plans to move forward. The brain has to drive musculoskeletal system to make the animal walk. But the heart rate will typically go up. The digestive system and even the immune system will be modulated, and these systems will signal back to the brain. So again, this forms a loop between the brain, the body, and the environment that as scientists we have to understand. We have to understand biological systems of many interacting elements, both between animals and their environment and within animals. And the challenge is to identify distributed control systems and also to make it more human understandable, to identify the elements within these systems with the most explanatory power for the phenomenon we're studying and the strongest causal relationships with it. So in the lab, the eventual goal is to achieve a theoretical understanding of these systems using tools from machine learning and neural computation and others. And so over the years, we've developed techniques. We and others have developed techniques for accessing different components of this system in larval zebrafish, which are transparent and small, and genetic tools are available. And at 7 days, they already exhibit a range of interesting behaviors, including learning, memory, and sleep. So of these different technologies, I will explain several of them during this talk. But I would like to start with a biologically question-driven set of results, which have to do with a behavior that animals use presumably to stay safe and also to eat. So we call this, this is known as the optomotor response. Also, Mike Orger's lab has achieved a series of crucial insights into this behavior. And there are multiple interpretations for why animals do this. They stabilize their location in space approximately over time by swimming in the direction of optic flow, which corresponds to the opposite direction to water flow in real environments. And one reason probably is to stay safe, because if they wouldn't do this, they might be swept into areas of unknown predators and maybe even dry land. There are various other reasons they might do this. One is that this allows them to take in food that flows along, small organisms that flows along with the water flow. But there's a trade-off between doing this and preserving energy. If animals do this continuously, they will consume a lot of energy. It's kind of a treadmill for fish. And running out of energy is itself a threat to health. And sometimes it's better not to keep doing this, but to, in some sense, give up on trying to perform this innate behavior. And this might be the case when the currents are too fast or chaotic to have any hope to stabilize their location. Similar behavior may also happen when animals are stuck in debris trying to free themselves. If they really can't free themselves, it might be better to stop trying and preserve energy and save themselves from injuries. Or when they're in low temperature water, when the muscles are less effective, it may be inefficient for the brain to keep trying to move when the muscles don't allow for it. So this is old work by Yu Mu, a very talented postdoc who's now a faculty at the Institute of Neuroscience in Shanghai. Again, this is published, but I'm showing you this as a background to newer work. So in the lab, we often use virtual reality environments where in the real world you might imagine that water flow moves the animal backwards, which the animal counters by swimming forward. In virtual reality, we head fix the animal, we move the ground in the opposite direction, so the ground moves forward when we simulate backward bodily motion, and when the animal swims, the visual stimulus gets accelerated backwards. Now we can take this one step further and paralyze the animal, which stabilizes its brain for imaging and also has other advantages. And we use electrodes to record non-invasively from the tail. So we record motor nerve impulses through the skin, and we can decode these into intended locomotion or swim attempts and give the animal realistic feedback in response. And we call this fictive swim bouts. So this is a preparation we often use, and it allows for naturalistic behavior like this animal performing the optomotor response in 2 dimensions. So it's swimming against a simulated water current. It's simulated purely visually by moving the whole environment in the opposite direction to the simulated current. So that the fish is there. It's only 4 millimeters long and it's quite transparent, so it's hard to see. So we pair this with whole brain light sheet imaging. So this is the virtual reality setup. We pair this with whole brain light sheet imaging that you also know from the Orga lab here, which is an adaptation that we made in 2013 from light sheet technology that we adapted for use in larval zebrafish to allow for imaging almost the entire brain in single cell resolution at several brain volumes per second, which is fast enough to track calcium signals using genetically encoded calcium sensors. So, and besides calcium sensors, there's also, of course, all the other sensors that are available. So coming back to this behavior, we label this positional homeostasis when animals try to approximately stabilize their location in space. And we call this in a VR setting closed loop because there's a loop between motor output, a change in position, and visual feedback. If we open the loop, in some sense, this becomes a helpless environment where animals can try to swim, but nothing happens. This we call an open loop environment. It's illustrated here. And as a result of several seconds or tens of seconds of swimming in open loop in this futile environment, the animal tends to give up, which is quite a familiar behavior that we would probably also perform if we were in the same situation. So we call this futility-induced passivity. Here is an example data set from an example animal. This is swimming closed loop where they swim against virtual water currents. They swim in discrete bursts, discrete bouts, about once per second. You can see that in open loop, they start swimming with more variability, more amplitude differences, and more turns. So remembering closed loop swims are effective, in open loop swims are ineffective, and that results after, on average, about 20 seconds in the animal to give up. And this is what we call futility-induced passivity, because it's behavioral passivity in response to behavioral futility. If we keep the animal in an open-loop environment, they'll switch between active and passive states as they're making attempts to move, giving up, waiting a bit, and trying again sometime later. We see this type of behavior in many other species, in many other situations, like this squirrel trying to get to a food source, but the pole is slippery, so you can see the sudden transition from a vigorously active to a sudden passive state. The animal doesn't fall asleep, it's still looking around and doing things, but it's given up on this particular behavior. And the dynamics are quite similar. There's a period of vigorous attempts followed by a sudden switch to behavioral passivity. So if we consider control theory account for this, normally a swim would lead to a change in position. But in these cases, the behavior showed that the brain of the animal can register when this fails, when there's no change in position, and then evokes an integration, which can be seen as a counter or a temporal integration that counts the number of failed attempts. And when this crosses some critical value, which we can model as a threshold, where there are too many failed attempts, behavior is inhibited for some period of time. We can use this control theoretic model to then study the brain and see whether we can identify the different components of this model in brain activity. And here you can see a data set where we image neurons and also astrocytes in the brain, so a non-neuronal glial cell type that we had to image in order to be able to explain this behavior after spending almost a year studying only neurons. So this is whole brain cellular imaging. I'm going to show you this example data set as an introduction to the next part of the talk. So this is swimming. The swim signal, the electrophysiological swim signal shown at the bottom. It's going to scroll to the left. The yellow bar is the current time. The movie sped up by a factor of almost 3, and neurons are shown at the top, astrocytes at the bottom. And here you can see when animals in a state of active swim attempts, there's a lot of neural activity, but when the animal is in the state of passivity, there's more radial astrocyte calcium signals than during activity. But importantly, the radial astrocyte calcium signals already start rising during these vigorous swim attempts, as you can see here, and then are maintained during the period of passivity. And so, if you take snapshots during this whole process, you can see that during, these are real snapshots, the green line is a cartoon, but during closed loop, radial astrocyte calcium levels are relatively low. During open loop, they start rising or integrating behavioral failures, and roughly the time when they peak is when the animal gives up, and those calcium levels are maintained at high levels for about 10 or 20 seconds during the period of passivity. So from this, we conclude that astrocytes integrate swim failures. Of course, we did more detailed studies of behavior and these signals together as well to back this up. Then Yumu performed a series of causality experiments where he activated astrocytes using chemogenetics and optogenetics to show that this induces behavioral passivity. And when he silenced astrocytes using a variety of methods, including pharmacology and cell ablation, then those perturbed fish almost never give up, and they persevere even when swim attempts are failing. So we conclude that astrocytes trigger the transition to the passive behavioral state. Then we could boil down to an essential circuit in the brainstem, which consists of 3 cell types: noradrenergic neurons in a brain area that's called the noradrenergic cluster of the medulla oblongata, which we call NEMO for short. This is in the nucleus of the solitary tract. And then astrocytes, which are functionally connected to these neurons, also with through anatomical proximity in a specific area of the brainstem in the lateral medulla oblongata, and that also contains GABAergic neurons that are shown here in blue. So A simplified cartoon information flow occurs as follows. So during behavioral failures, noradrenergic neurons detect the coincidence between swimming and the absence of expected feedback. So this failure signal then gets integrated by astrocytes through noradrenergic signaling, and when this crosses the critical calcium concentration, this triggers activity in GABAergic Elmo neurons that then suppress swimming. And in this case, this is not always the case, of course. A lot of neural computation occurs through distributed processing, but in this case, we were able to map these different components of the control theoretic model to different cell types, as you can see here indicated by color. Now, after this, there were a variety of other questions that we asked following this deepened understanding of the role of astrocytes in neural processing. There were open questions about how the different cell types communicate with one another at the molecular level. So we already understood some pieces of this, but a crucial piece was not understood. So what we did understand was that norepinephrine, this norepinephrine signal from the failure-detecting neurons gets received by astrocytes through the alpha-1b noradrenergic receptor, for example. That was also expected from previous literature in mammals. But what was unknown was how astrocytes signal back to neurons through a process that some people may call gliotransmission. There were many questions and many hypotheses about how this might work, but not a good understanding about how it does work in vivo. So this is new work by Alex Chen. That is a very talented graduate student, jointly supervised with Florian Engert at Harvard, which is the lab where I did my postdoc. So Alex asked, how do astrocytes signal to neurons? And after a lot of exploration, converged to studying the pure-nergic system, which is based on ATP and adenosine signaling between cells. So ATP is most well known as an energy currency used by cells across the body, but it's also a neuromodulator. So in this data set, you can see astrocytes intracellular calcium signaling during the futility assay that I just explained, together with radial astrocyte extracellular ATP signals that Alex imaged using grab ATP from Yulong Li's lab. The signal in the ATP channel is not as strong, it's not as clear because it's a new sensor that's not as developed as GCAMP. But if you look at the average signal, you can see that calcium and ATP are strongly locked to each other during futile swimming. They rise together, they peak together, and they decay together, but you can see that the ATP signal has a bit of a delay relative to calcium, which hints to a model for glia to neuron communication. We call this model 1, where astrocytes signal to neurons by secreting ATP when calcium levels are high. ATP binds to ATP receptors on neurons and activates spiking in those neurons. So Alex set out to test this model. And here you can see a data set where there's a control trials shown in green. This is the expected case. The ATP, extracellular ATP levels are shown on the y-axis. And this is the same kind of data you saw before. When animals perform futile swimming, this is a series of futile swims. ATP levels rise in the extracellular space. So that's the normal case. Now, if you block norepinephrine receptors on astrocytes using pharmacology, you don't see this rise in extracellular ATP, which is a hint that astrocytes are releasing ATP. To be even more specific, you can suppress glial calcium levels using a tool developed by Bal Kak's lab at UCLA that pumps calcium out of astrocytes and prevents calcium levels from rising too much. And in this case, you also see a lack of ATP in the extracellular space, from which we could conclude that astrocytes are necessary for ATP release. And specifically, calcium level rises in astrocytes are necessary for ATP release. Alex also did the opposite experiments to show that if you anesthetize the neurons, you block sodium channels, so you prevent spiking in neurons. If you induce calcium levels to rise in astrocytes, you get an increase in extracellular ATP, showing that they're also sufficient. So the next question was, what does ATP do to behavior? And for this, Alex used caged ATP and uncaged it in the brain. And as expected from model 1, so that there was an increase in behavioral passivity. That's consistent. If ATP is the signal that travels from astrocytes to neurons, then artificially increasing ATP in the extracellular space should have the same effect as astrocyte signaling onto behavior. So that's consistent with model 1. But then came the confusing piece of data that was inconsistent with model 1. If you block ATP receptors, then you don't get a decrease in passivity. So if you block ATP receptors, you'd expect to see the opposite of what you see after ATP elevation, but this didn't happen. So ATP receptors are not necessary for passivity. So that's inconsistent with model 2. This led to the next model that's based on the observation that ATP is crucial, but ATP receptors aren't involved. So ATP doesn't just remain ATP in the extracellular space. It's actively metabolized by two enzymes into adenosine. And so the alternative hypothesis was that it's this biochemical cascade in the extracellular space that's causal to this behavior. So there are two enzymes, CD39 and CD73, that convert ATP into ADP, AMP into adenosine. And neurons are also known to have adenosine receptors. Now we can interfere with this pathway through two compounds, ARL67156 and AMPCP, which compete with these enzymes for, or that compete with ATP and with AMP for these enzymes. For example, the first one is a non-hydrolyzable ATP analog that binds CD39 so that less CD39 is available to convert ATP to ADP. So we can use these as tools to test causality in the extracellular space. So Alex found that if you block adenosine receptors, this reduces futility-induced passivity. So this is now the expected result that's consistent with this model 2 that involves adenosine signaling. You can see that there's a reduction in passivity if you block adenosine receptors, suggesting that adenosine receptors are indeed the recipients of the glial signal. Now, if you reduce ATP to adenosine conversion using these two compounds that I just explained, this also reduces futility-induced passivity, as you can see here. In both CD73 and CD39 blocks, you see a reduction in passivity, which in both cases is consistent with model 2. And so now I'm going to walk you through what we initially thought was the circuit that mediated this kind of behavior. Many years ago, before we started on this work, we thought that futile swimming in this helpless environment would lead to integration of behavioral futility. That's the coincidence between actions and the lack of expected feedback. That this might be integrated in recurrent neural circuits, as has been shown to be the case for a variety of tasks. So we thought it might be the case here. Integration of information in recurrent circuits that implement attractors or approximate attractors, where network activity integrates behavioral futility, and when it reaches a threshold on the order of perhaps 10 seconds, this may pass through a thresholding nonlinearity, which inhibits swimming and causes the fish to give up. So this kind of purely neural-centric model turned out to be wrong, and instead we need to look at astrocytes and pure energetic system, extracellular biochemical reactions to explain this, where futile swimming leads to the activation of noradrenergic neurons, the release of norepinephrine that activate astrocytes through alpha-1b receptors. Astrocyte calcium is the actual integrator, not network activity, that integrates the futility signal. And when this reaches a critical value on timescales of 10 seconds or longer, this releases ATP that gets metabolized into adenosine, which gets received by neurons, which then inhibits swimming through GABA receptors, leading to behavioral passivity. So this kind of really broadened our picture of the types of systems that implement these types of behaviors. So I'm going to, in the interest of time, I'm going to skip through this other summary. This is basically the same as what I just explained. But the other way to view this is that this system implements a kind of very slow temporal filtering operation, where there's an excitatory phase where norepinephrine excitation wins over purinergic suppression, and then an inhibitory phase where noradrenergic excitation loses over purinergic suppression. And this kind of temporal relationship is able to implement this this period of vigorous behavior followed by a sudden switch to passivity. So to summarize again, our understanding broadened from a purely neuronal system to a system that's neuronal, astroglial, biochemical, and neuronal as an implementer of these internal state switches. Just to summarize it, we found that astrocytes integrate behavioral failures over time and trigger brain and behavioral state switches. And that this relies on communication between the noradrenergic system, astrocytes, ATP, adenosine, and adenosine receptors, and that astrocytes, like neurons, are computational elements in circuits and can control behavior on short timescales. So I just described how this all leads to behavioral switch, but so far we've used behavior as a readout. But the next question was, what happens to the entire brain during this process? So how does the internal state change impact brain-wide neural computation? So you might also have noticed that I switched from calling it a behavioral state to calling it an internal state, which I'll explain to you in a second. So what we wanted to do is to expand the assay that we just used. So where previously we contrasted closed-loop swimming with open-loop swimming. In closed-loop swimming, we have swim attempts that lead to forward displacement of the animal. And this doesn't lead to an internal state switch because this is the standard way that animals interact with their environment. So following this, we call this now an engaged internal state, engaged with visual flow stimuli, because they still interact with visual flow stimuli. And then we used a pulsatile motion integration, where we have the same visual environment that moves forward, but in short pulses, slow forward motion in 1 second pulses interspersed by 1 second of silence. And in this way, we could look at how the visual system and other systems integrated this visual motion over time in discrete chunks. So we contrasted this with open-loop swimming that I just described, where fish swim in a futile environment, where swim attempts don't lead to any movement. This leads to a state switch to futility-induced passivity. We call this now a disengaged state because animals stop interacting with their visual environment for some period of time, where slow visual pulsatile motion doesn't immediately lead to swimming. So more specifically, we call this effective trials, where effective swimming leads to or maintains the engaged state. Just to walk you through this, we have an evoke epoch where closed-loop swimming leads to visual feedback. And now we have a pause epoch where we simply stop moving the stimulus forward, so the animal has less reason to swim because there's no observed flow anymore. The fish typically stop swimming for some time, and then we show the animal these slow visual motion pulses that the animal then integrates over time and eventually swims. So we call this the engaged state, but crucially in the engaged state, the animal is not swimming for some period of time, but the brain is still integrating this visual motion. We can now contrast this with futile trials where we have the futility assay that I just described, and there's one difference with what I just showed you, that there's an open loop period where swimming now doesn't have any visual consequences and triggers, so this futile swimming now triggers futility-induced passivity. When we detect automatically during the experiment that the animal has switched to this passive state, we trigger a pause period. Sorry, we trigger the pause period, followed by the probe epoch, where we show these forward pulsatile motion stimuli. And again, the animal is not swimming, but it's receiving and integrating these visual motion stimuli. And we can now study how the brain integrates this exact same signal under the same behavior, namely no behavior, but in a different internal state. So we find that on effective trials, the fish swims earlier than on futile trials, which is consistent with our previous expectation of futility-induced passivity. Now we can try and decompose what happens in the brain in terms of discrete processing stages. And this is, of course, a cartoon. Here we assume a conceptually simplified model where the expected dynamics is shown for the different processing stages, starting with sensory encoding, sensory integration, premotor, motor, and swimming. So we assume that sensory encoding simply represents the stimulus. Sensory integration integrates this over time. This is now a non-leaky, idealized integrator. Premotor signals should come on sometime before swimming. Motor signals should come on near or at the time of swimming. So we can form several hypotheses about how this internal state switch may change these different signals in the brain. So you can see some of the hypotheses where sensory encoding may be dampened, or sensory integration may be slowed down, become leakier and less efficient, or swim thresholds may go up. Maybe the animal needs to receive, maybe the motor system needs to receive more drive before the animal can swim, or maybe the connectivity between these, the functional connectivity between these different processing stages is weakened. And again, this is of course a very simplified model, but it allowed us to decompose the data into different pieces. And here you can see that decomposition represented in different brain maps, showing you neurons that respond to visual motion, for example, here on the left, neurons that integrate visual motion over time, and so on. I want to focus on just a few of these stages. Here we can see functional signals corresponding to them. So you can see the sensory encoding stage. In black is shown the normal internal state, or the engaged internal state. Here, neurons respond to the visual motion pulses reliably, but in red is shown the disengaged state. You can see that there's a small reduction in amplitude of these responses. And this is also consistent with recent work from the lab of Germán Zumbre by Alejandro Uribe Arias. Now, in the sensory integration stage, you see the biggest difference of all. Here, in black, you can see the integration in the engaged state, and in red, you can see the integration in the disengaged state, where the integration is far less efficient and almost flat. So it's the amount of integration, or the leakiness, is changed by a factor of more than 7. This is a piece that I'm going to skip. This is the premotor and the motor signals, where you simply see a shift to a later time in correspondence with behavior. So I really want to focus on the largest effect that we saw, that was at the integrative stage, where you can see that the amount of integration is vastly reduced in the disengaged state, which suggests that this change in internal state most strongly impacts the intermediate stage of processing between sensation and action, suggesting that this is of course not always true, but some internal states may affect these intermediate processing layers the strongest. So from this, we conclude that the futility-induced disengaged state can change not only motor output, but also brain-wide computation, and that the most strongly affected processing stage is motion integration, which becomes roughly 7 times leakier and less efficient. So next we asked whether astrocytes are responsible for this brain-wide change in dynamics. We know it's responsible for the change in behavior, but we didn't yet show how it affects the rest of the brain. This is just to control for that astrocytes show the expected dynamics. This is astrocyte calcium levels, where you see on futile trials calcium levels are enhanced, but in the probe period, before animals start responding to the visual stimulus, astrocyte calcium level has decayed back to baseline. Okay, now we can use a tool from Balcock's lab that we used also before, called CalX, to silence astrocytes by pumping calcium out. And in this case, here you see the normal case, where you see a small reduction in the sensory representation and a very strong reduction in the integrative component of the response. This is in intact astrocytes data you saw before. This is now with astrocytes silenced. The difference between the 2 different trial types, where the animals either experience a closed loop or an open loop environment, go away. So you can see this also reflected in the amount of integration. The integration now becomes similar between the 2 internal states. So we conclude that astrocytes drive this brain-wide change in the dynamics, and this may potentially also happen through ATP, but this needs to be tested because it may depend on the brain area. Even though we showed that ATP to adenosine conversion is necessary for the final motor stage, we still need to test whether there are brain area differences, where there may be additional pathways involved in, for example, the visual system. So I now want to change gears to a more technical component of what the lab does. So in the lab, we often iterate between technology development and biological question-driven research. And in what I just showed you, there's a utility of combining modalities. We're broadening from neurons to neurons and astrocytes enables new types of questions to be asked and addressed. We wanted to broaden across other modalities as well. And as you saw, the whole brain light sheet imaging is good for screening neural activity across all neurons in the brain, but what's missing is connectivity. So we next wanted to merge whole brain activity, connectivity, and behavior by performing functional imaging followed by whole brain connectomics. We think this will open up new opportunities for discovery through machine learning, computational neuroscience, biophysical network models, and other approaches. And so the idea is to perform functional imaging. This is just a cartoon, followed by the reconstruction of connectivity across the brain. And this is led by Alex Chen, joint student with Florian Engelt's lab, and Mariela Patkova, joint between Florian Engelt's lab and Jeff Lichtman and others. So Alex did functional imaging of the types of behaviors that I just showed you, plus 7 other behaviors, visually driven behaviors, like phototaxis, looming avoidance. And this is in order to create a community resource that allows for many questions to be addressed in collaboration with Janelia EM, the Florian Engelt's group, Jeff Lichtman, Vera and Jane at Google, Janelia project team, and funded by Simon's collaboration, NIH, and HHMI. And this is now a functional data set showing you a very coarse clustering of neural activity, so cells by time. You can map these clusters to the brain to start seeing anatomical localization, but this is really the crucial piece. This is the same animal that's been functionally imaged on the left and anatomically reconstructed and segmented on the right. Across the entire brain, and so we can map this activity to connectivity to each other at the single cell level across the brain. So right now, about half of the cells are registered. The goal is to get this registered for almost all the cells. And here's now just some of the neurons that are reconstructed through a proofreading team based at Janelia. Showing you a subset of neurons reconstructed and selected for symmetry across the brain. And this eventually will allow for the joint study between activity and connectivity, as you can see here, for tactile neurons represented through their morphology over here, and activity of the tactile neurons here in black, the input neurons in green, and output neurons in blue, together with these different behavioral assays consisting of visual input that drives different types of behaviors. The goal is to create a resource where, by virtue of these multiple behaviors, that the community can ask multiple questions and look how the system as a whole allows the animal to generate behavior from sensory input, and also through feedback from its own behavior. We're also combining modalities across other components of the biology, specifically the biophysics and molecular signatures of individual neurons, by combining activity, behavior, and transcriptomics through advances in tissue processing, expansion microscopy, and also computational methods for single cell registration, transcript assignment, and eventually multimodal analysis and modeling. So this is work by Emmanuel Marcus Legoretta and Greg Fleischmann and many others, where we're combining neuronal activity during behavior. The behavior here is represented by this, the cartoonized activation of muscles in the spinal cord, and the expression of genes across the brain visualized through hybridization, chain reaction. We are recovering about 40 genes per brain at the moment, and these are superimposed at cellular resolution across most of the brain. So we can now also combine brain activity, behavior, and the molecular signatures of neurons, which could be used for cell typing or for biophysical understanding of the cells through, for example, peptidergic communication pathways or spike generation processes. Okay, so the final modality that we want to combine with brain activity and behavior is the rest of the body. As you heard from the organizers, the body is a crucial component in the way that the brain interacts with its environment. The environment is both the outside world and other organisms in the environment, but also the body itself. And so this is again in the theme of non-canonical substrates of computation. Perhaps there's biological computation that occurs beyond the brain in the nervous system throughout the body. And of course, Carlos Ribeiro is a pioneer in studying these systems. And so life consists of this elementary functional unit, the cell, or depending on who you ask, I think it's the cell, is a good way to start. But these cells are networked across the brain and the body. And so Virginia Rutten, who's an extremely talented and creative student, now a postdoc in the lab, expanded from the brain to the rest of the body to include the peripheral nervous system, but also other tissues, because it's not just the peripheral nervous system that's capable of performing computation, but also cells that may not be neurons, like astrocytes in the brain. Maybe there are other cell types across the body that partake in neural computation or in biological computation. And so that's why Virginia, in collaboration with others, considered body-wide control systems and wanted to include not just the brain, but also other pieces of the body, many of which are shared with us as fellow vertebrates. By combining these three, in order to combine these three, Virginia managed to create a transgenic fish with calcium indicated in all cells in the body instead of in all cells in the brain. And here you can see this animal labeling cells across organs and tissues. Here's a fly through this animal. There are various versions now that we've made with various efficacies of labeling and calcium reporting. And I should emphasize that calcium is just a starting point for this, because of course cells, particularly outside the brain, calcium is just one of the indicators of what the cells are doing. We probably need to combine this with many other imaging modalities for cyclic AMP, ATP, extracellular peptides and hormones, and so on. So, but calcium is a good start because cells use calcium for signaling of many processes. Here you can see a variety of organs and tissue types, muscle, skin, brain, liver, and so on. You can already, from the cell size and shape and tissue texture, tell what these are, but of course we want to look at dynamics. So this is now a dynamic signal because it's GCAMP. Here you can see whole body cellular dynamics. This is sped up by a factor of 50. Things do happen more slowly on average in the rest of the body compared to the brain, but some processes in the peripheral nervous system and muscles, for example, are almost as fast. So here's an annotation. You can see the brain, muscle, see the aorta, kidney, and the gut, together with many other tissue types. Here's now another view where you can see the brain more clearly. I believe we can identify neuronal and astroglial processes in the brain because this is a ubiquitous marker, as well as signals in the skin, muscle, and other tissues. Now, the tissue outside the brain is a lot more, moves a lot more. You can see that here. So together with Wei Zheng and Guosheng Yu, who are engineers, they created an optic flow-based registration algorithm that can take care of these large movements reliably and register tissue so that we can specifically disambiguate calcium signals from tissue motion. And so that eventually gives us a data set consisting of cells across different tissue types in the body and across cells and across time. We can now cluster this, so Virginia created a coherence-based clustering algorithm that clusters these signals into time series, which we can then map back to the tissue to anatomical space to start manually identifying what these different tissue types are, mapping across them, and then also using that to give labels to these time series. Again, as you can see, the skin, the kidney, the brain, and as one example here, we can see tissue subcompartments in the kidney. So these are dynamically defined subcompartments, where there are clusters of neurons that do something similar across time. So you can see that here, these are the subcompartments that you can also temporally order and that reflect this temporal dynamics in the kidney that was previously unknown to exist. Here you can see pulsatile waves traveling across the different compartments, here more specifically shown as a matrix where you can see pulsatile dynamics that sometimes get transmitted to the next compartment of the kidney, and sometimes there's a failure in transmission. This is calcium, so we don't know yet what the purpose of this is. Maybe it produces fluid motion through osmotic pressures. We don't know that yet. This is still to be clarified. Now, this is dynamic data. We want to map that to anatomical data as well to match that with a cellular understanding of what's underlying it. So together with Paul Tilburg and Mark Edison and Amy Hu, Virginia created a whole body expansion protocol so that these signals can be matched with their anatomical and molecular underpinnings. As you can see here, this is the peripheral nervous system, including here the enteric nervous system. Together, we call this system holistic, which is also an acronym. Here's one example study where Virginia looked at whole body coping with hypoxia, where we lower the oxygen concentration in the bath. She looked at calcium signals across the body and also movement, and found that there are specific cells that are activated by hypoxia, which in this case is part of the sympathetic nervous system. She also found a concurrent narrowing of the mesenteric artery, which feeds the digestive system, and this had an effect on blood flow. As you can see here, if you look at this square, less blood is delivered to the gut during hypoxia, and this may be a mechanism for actually making the gut use less energy resources when energy is scarce, and preserve energy resources for more critical organs like the brain. Here, the putative models, the brain drives activity in the sympathetic nervous system that causes narrowing of the mesenteric artery, causes reduced blood flow to the gut, which may then lead to reduced gut activity that is simply due to the less energetic resources available, although it's possible and likely that the nervous system also directly inhibits the gut. So this is just one of several findings that Virginia made using this system, and verified through optogenetically inhibiting the brainstem, in which case you can see that this is now the normal case, the non-inhibited case, where hypoxia leads to a drop in mesenteric blood flow. But if you, so this is the control case, but now if you inhibit the hindbrain, then blood flow returns back to normal levels, suggesting that the brain is indeed driving this process that leads to less blood delivery to the gut. Together with Chie Sato, another group leader at Janelia, Virginia's been generalizing this to adult vertebrates, namely Danionella, that you also know from work here in the Orger lab. And here's just an idea of what the kinds of signals you can get in the skin of Danionella. These animals are harder to access in deeper layers, so this is still a work in progress. So I want to just close with also again echoing the introductory remarks that I think that combining different disciplines is both necessary and both a luxury for neuroscience. I think it's necessary to be able to understand these distributed systems from the cell biological level to the whole organismal level. And it's also, I think, a privilege to be able to talk to all these different communities and learn about all these different fields. So finally, thank you to the people who carried out this work, lab members, former lab members, institute staff, and collaborators, especially Virginia Rutten, who did the pioneering work on whole body imaging that you saw at the end, Alex Chen, who worked on astrocyte to neuron communication, building on work by Yu Mu, who did the initial work on filtrate-induced passivity. And then Jingxuan Lim, a student who graduated, who worked with a postdoc, a very talented computational postdoc, Xiqiang Wei, on the computational side of the whole brain modulation. And then also Mariela Petkova and the Google team and Harvard on electron microscopy, and other collaborators who worked on various components of this research. And thanks for listening.
Okay, so we have about 10 minutes for questions.